Imagine while baking a pizza, you accidentally use chocolate sauce instead of tomato sauce. The end product won't be a pizza, but a new dish. Likewise, in your body, if a particular base in your DNA is inserted, deleted, or rearranged, it would result in a mutation, which can be defined as a change in the genetic code resulting in a loss or gain of a codon. Mutations can be triggered by a host of factors, including physical and environmental factors, such as exposure to the ultraviolet rays of the sun, radioactive radiations, or harmful chemicals. Mutations, which often result in undesirable effects, can be classified into various types, including point mutation and frame shift mutation. To understand these two types of mutations, let us use the statement, "The cat ate the rat," to represent an DNA template. The individual words of the statement represent triplet DNA codons, and the letters represent nucleotide bases. Moreover, just like an DNA template, the statement is a reading frame where the words are contiguous and non-overlapping. In point mutation, only one base is substituted by another base, which results in a change in the genetic code. In our statement, if we replace C with B, the altered statement will read, "The bat ate the rat." Such a change in the genetic code can lead to undesirable consequences. For instance, a change in a single base pair in the gene for the beta-globin chain results in a change in amino acid residue glutamate to valine, which ultimately leads to sickle cell anemia. In the case of frame shift mutation, two or more bases are either inserted or deleted from the DNA template. For instance, in our example, if we insert two letters R and K, it will change the reading frame, and hence the altered statement will read, "The cat ate her at." Whereas, if we delete the two letters, that is one C and one A, from the original statement, the altered statement will read, "The tat eth era T." Likewise, in a frame shift mutation, if three or multiples of three bases are inserted or deleted, it will result in the deletion or insertion of a codon. This in turn will result in the creation or deletion of an amino acid. Interestingly, frame shift insertion or deletion of one or more bases changes the reading frame only from the point of insertion or deletion. This point is clear when we compare the original statement with the statements that represent the mutation. Did you know that frame shift mutation is common among textile workers? This is because they are often exposed to acridine dyes such as 5-aminoacridine, which enters their bodies through inhalation or physical contact. These dyes get intercalated or wedged between two adjacent purines and thus increase the distance between them from 3.4 angstroms to 6.8 angstroms, leading to frame shift mutation. Chemical, physical, and environmental factors thus lead to the addition or deletion or replacement of one or more base pairs of a codon, which in turn causes frame shift mutations and point mutations. A series of experiments conducted by scientists and biochemical experts proved that both RNA and DNA served as genetic material. However, the question that arose was, what exactly made both DNA and RNA suitable genetic materials? The answer lies in the fact that for a molecule to be a genetic material, it must fulfill four simple conditions. It should be structurally and chemically stable, which means it should not change with age, change in physiology, or life cycle. Yet, despite stability, The molecule should provide scope for slow changes or mutation that is necessary for evolution. Further, the molecule should be able to express itself in Mendelian characters and also be able to generate its own replica to become a genetic material. Most biomolecules, including proteins, fail to fulfill these criteria. Even so, both the nucleic acids, DNA and RNA, meet all these requirements. Although it is to be noted that DNA serves predominantly as genetic material, RNA also acts as genetic material in few microbes such as tobacco mosaic viruses and QB bacteriophages, and also performs the role of a messenger and adapter. Of the two nucleic acids, it was DNA that played a part in Frederick Griffith's experiment, where R-strain bacteria had been transformed to living S-strain. However, though the transformation killed the mice, it had not changed the properties of the DNA. This can be explained by the stable structure of DNA that, although being separated due to exposure to heat, it can come together under suitable conditions even after. RNA, on the other hand, is much more liable to change due to the presence of the very reactive 2-OH group that is present in its every nucleotide. This makes RNA more degradable and reactive than DNA. Also, it enables RNA to act as a catalyst in several biochemical reactions in living systems. Moreover, DNA has thymine instead of uracil that is present in RNA, which makes it more stable. For this reason, it is also said that DNA has evolved from RNA with chemical modifications, making it more stable and, as a result, a better genetic material. Yet, both DNA and RNA can mutate. However, viruses with RNA genome evolve faster by mutation due to the ability of RNA to mutate quickly on account of its relative instability. Nonetheless, RNA can directly code for protein synthesis and thereby easily express the characters. In this regard, DNA is dependent on RNA, not only protein synthesis, but also several essential life processes such as translation and splicing are evolved around RNA. Therefore, even though DNA is the preferred genetic material due to its stability, it is the RNA that is involved in the transmission of genetic information. Proteins play a pivotal role in the proper functioning of any organism. The synthesis of proteins occurs in a cell in two phases, namely transcription and translation. The process of converting mRNA codon sequences into an amino acid polypeptide chain is called translation. Prior to translation, mRNA, which is synthesized in the nucleus of a eukaryote cell, migrates through the nuclear pore into the cytoplasm of the cell. The cytoplasm also contains tRNA, amino acids, and ribosomes, all of which are involved in the process of translation. The amino acids and tRNAs in cytoplasm become activated by a process called the charging or amino-acylation. During this process, amino acid binds with ATP in the presence of amino-acyl RNA synthetase and is activated into amino-acyl AMP enzyme complex. This complex reacts with tRNA and transfers the amino acid to it. As a result, tRNA becomes activated as amino-acyl tRNA and the enzyme and AMP are liberated. The tRNA has an anticodon loop which determines the amino acid that will link to it. This anticodon of tRNA is complementary to the codon sequence on the mRNA and will only bind to that codon. Apart from tRNA, a ribosome is another component required for translation. It is made up of structural RNAs and about 80 different proteins. In the inactive state, a ribosome consists of two subunits, a large subunit and a small subunit. The large subunit consists of two sites, the P or peptidyl site, also called the donor site, and A or amino-acyl site, also called the acceptor site. At these sites, the two subsequent amino-acyl tRNAs bind and facilitate the formation of a peptide bond between the two amino acids. The ribosome also acts as a catalyst in the formation of the peptide bond. Another component in translation is mRNA, which consists of the start codon AUG that codes for methionine, the stop codon, which is either UAA, UAG, or UGA,